Okay. So, a very good morning to you all, and welcome to the third day of our 5 day long workshop on next generation big data biology. Yesterday, we have heard a very detailed protocol and procedures on this DNA extractions and different types of problems, different types of advantages and disadvantages on this protocols. And today we are expecting to hear something more about the topic. So without wasting time, I am inviting Mr. Shubhrajit Roy to continue his discourse. Mr. Shubhrajit Roy, please.
Thank you, Dr. Rai. Uh, I'll be sharing my screen. So is it visible?
Yeah, it is visible.
Okay.
Right.
So let's start with today's discussion. Good morning, all of you. And yesterday I have discussed about how to extract the genetic material from the environmental sources for the purpose of metagenomic metagenomic library preparation. So uh, you must be wondering why I have used the term library here. As you can see in my first slide, I have put an image of a library behind my title, as it has nothing to do with whatever we are going to discuss today. So library, the image is, as it shows here, is a collection of books, actually books, audios, films, whatever the documents, manuscripts were documented. Actually, we have lost our habits due to improved digitalization, that we frequently do not visit library for our study purposes. So if you have an habit, particularly the students who have are studying, if have a habit to visit a library, they will find it's a curated collection of different sources of information. And when you go and ask the librarian for any book, he usually look for the particular number from any shelf, and then you can easily find the book. So my purpose of explaining this library thing to you is because of the fact that it's because it's a very organized system. Library acts as a very organized system, and it is easy to find any book because of certain tags or barcodes which has been provided to the books. And you can easily identify any particular book or material from that collection using that uh, barcode. So in this metagenomics, the library, the term is the purpose of the library preparation is to introduce an adapter sequence. These are nothing but oligos which are introduced to the template molecules. Template molecules generated because of the fragmented genetic material, and then which allows the amplification of individual templates. If you are using any sequencing platform, I will discuss in my next talk tomorrow. So you will find that these platforms are provided with short oligos which are complementary to these adapters, and using these adapters they can sequence the fragments in individual fragments of this DNA. So classically, a metagenomic library preparation includes these 4 steps. So first one is the isolation of the environmental DNA, which I have discussed in my last talk. Second one is the choice of an appropriate vector. So before you need to clone this fragmented genetic material into any vector. And next one is cloning for the library construction and screening of the further DNA analysis. Well, we do not practice these kind of library construction nowadays, but before any development of next-gen sequencing, even for the Human Genome Project, you will find that it has been in the use of appropriate vectors has been there, where the genetic material was fragmented and introduced into the cloning vectors for downstream sequencing and analysis. So for that classical approach, it includes the construction of small insert libraries. If you are interested in cloning small inserts that is greater than so less than 10 kb in standard sequencing vectors using E. coli as a host. On the other hand, small insert libraries do not allow detection of large gene clusters. Here is the importance of large insert libraries. The large insert libraries are cosmid vectors, which can have a capacity of intake of insert of 25 to 30 kb. Phosmid vectors are about 40 kbs, and bacterial artificial libraries are able to take about 200 kb of DNA fragment. So if I can segregate the long size and short size insert according to their purpose, each of the purpose is can be classified into sequence-based approach and an activity-based approach. So if for the short size insert size cloning vectors are for the normal plasmid vectors or lambda vectors, which include the plasmid blue script, general T vectors. And for the activity-based approach, we use PLAC vector or T7 vectors. For the long size insert, which can be up to 10 kb to 100 kb in size, the long maintenance insert vectors were used, like BAC, phosmid, and cosmid vectors. So for metagenomics can be classified into these two large and short insert sequencing library construction approach. For the large insert, this is for sequencing and pairing sequencing, generally for 16S rRNA gene and phylogenetic markers. And for the small size insert, we use to identify the genetic heterogeneity, molecular evolution, comparative genomics. These can be used for you through paired end sequencing and also functional screening for heterologous expression systems. So this is a pictorial representation of whatever I have discussed now. So if you can isolate the genetic material from any environmental sample and then ligate into the restriction digested vectors, like the vectors I have discussed here. And after ligation, these vectors were introduced into E. coli cells, and then E. coli cells will individually grow and contain that transformed DNA material inside it. And using those particular clone, either you can screen for the presence of any particular enzyme, or you can screen the genetic material and produce the contig and then go for assembly and annotation of the gene functions. So this is a detailed workflow of any sample collection and genetic material from the source. And if you are intended to form a plasmid based library preparation, you need to run those on an agarose block, and I digest those environmental DNA samples with particular enzymes, which will generate a blunt end or a sticky end DNA fragments. And those fragments were now inserted into bacterial artificial chromosome. Say, for example, you are digesting your genetic material with BAM H1 and digested the back vector with BAM H1, and then you can introduce these vectors and allow them to ligate, and these individual clones can be taken for functional screening, or it can be directly go for NGS analysis or sequencing. So the library preparation for any metagenomic analysis requires sampling and processing. So the DNA extraction should be representative of all the cells present in the samples. If you remember, I have told you in my last talk that in a metagenomic sample, there are different amount of microbial cells are present. So if you do not have any universal technique of isolating all the genetic material, and if that samples do not have a representation of all the genetic material from the cells, then your metagenomic approach may not be perfect. And the sufficient amount of high quality nucleic acids are required for the library preparation, because I will discuss in my next part that there are two approaches. One is PCR free and one is amplicon based or PCR based. So for the PCR based library preparation, you will find that the presence of any uh, inhibitors which can inhibit the PCR amplification can cause a huge problem for library preparation. So the sample fractionation step should be checked to ensure that sufficient enrichment of the target is achieved and that minimal contamination of the non-target genetic material. So physical separation and isolation of the cells from the samples might be important to maximize the DNA yield or avoid co-extraction of enzymatic inhibitors that might interfere with the subsequent processes. So some of the kit nowadays are provided with some cleanup reagents that can easily allow the inhibitors to be removed from the DNA sample so that the downstream mechanisms are not inhibited. So direct lysis of cells versus indirect lysis have an quantitative bias in terms of microbial diversity and DNA yield. So resulting in sequence fragment lengths. Some type of samples like biopsies, groundwater often yield very good amount of DNA, but in library production, most of the sequencing technologies require high amount of DNA, and hence the amplification of starting material might be required. Uh, I if I haven't discussed, I think I have need to discuss this, that metagenomics can also be applied for anthropological studies. So if you are interested in knowing the different timelines, if you are from any excavated material, maybe a bone, or uh, from the past uh, years, or to find out the evolution of animals, then probably the presence of microbes can help you to do that. So for those samples, they are not properly preserved. So the amount of DNA yield from those paleo samples can be difficult and require and may produce very small amount of DNA. And small production of DNA might not be very useful for amplification based library preparation. So though there are several advanced techniques are present, which is like MDA, which is multiple displacement amplification, or using random hexamers, or FAJ 529 polymerase, which is employed to increase the DNA yield. But even after that, the amplification bias cannot be removed from this metagenomics study. So as I have discussed, that library preparation can be of two different types. So if you are planning for a PCR based reaction, it is uh, generally cost effective but require excess amount of DNA for the purpose of creating the library. And of course there is amplification bias, because small amount of genetic material contributed by a very a small number of microbes may not be amplified and ultimately result in a community profiling where you are unable to identify them. So modern polymerases that are designed for complex samples like kappa hi-fi, NEB-Q5 or chiasic hi-fi polymerase, these are has been designed to amplify genetic material from very low input DNA. But even after that, we cannot remove the question of amplification bias. So the next one is option is PCR free technique, which is generally costly. And if you go back to the PubMed from 2001 2011, you will find that majority of the articles have performed metagenomic studies on PCR based library preparation techniques. So maybe because of the costs, and they are still believing on the PCR based techniques, and maybe the presence of modern DNA polymerases are helping them for the development of the library preparation. So if your experimental design is kind of surveying any particular area, say for example one site, you want to survey what are the microbial populations present in that, then 16S rRNA sequencing will be useful for you. So for the purpose of library preparation for 16S, it generates approximately 1500 base pair long sequences, and the 16S rRNA gene is, can provide 9 variable regions interspersed between the conserved region, and the primers are designed to bind to this conserved region and amplify the fragments. So the variable region of 16S are frequently used for these phylogenetic classifications, such as genus and species diversities in the microbial population. So as you can see in this picture, whether you are not being able to see it properly because it's very small. This is a typical workflow where we use 2 different forward and reverse primer for the amplification of this 16S rRNA gene, and then through some reduced polymerase chain reaction, we introduce the adapters at the 5' and 3' end, and then we can pull the libraries are normalized and pooled and sent for sequencing in different sequencing platforms. Apart from 16S rRNA, we are well accustomed with the term NGS, and NGS library preparation allows 16S whole identification of the entire genetic material. So for the purpose of NGS library preparation, we need to perform the DNA fragmentation and the target selection, adapter sequences ligation, size selection, and finally the quantification of the library and send it for quality check. So for the purpose of fragmenting the genetic material, so we need to, depending on the platform we are choosing, different platforms may have requirement of different fragment size. So you have to carefully select the sequencing platform and follow their instructions and what are the fragment size they require, and they prefer to use the DNA fragmentation techniques to allow their DNA products to run on that system. So for the DNA library construction, you need to fragment it. So the fragments may be generated using amplicons. So the using amplicons or random hexamers, you can identify or allow the amplification of certain regions of the entire genome. But there are certain areas which are consistently not amplified from the genetic material. So this is a particular drawback for the PCR based DNA fragmentation. And apart from the enzymatic fragmentation of the DNA, you can use different sets of enzyme to create a blunt end or a fragmented or a sticky end of DNA genetic material. And once these fragmented genetic materials are produced, the adapters are ligated in the 5' and 3' end and produce. Uh, if you are dealing with transcriptomics, where you initiate for the purpose of expression analysis, if you are starting with RNA, you can convert it into cDNA using RT-PCR and perform the same way by adapting by ligating the adapters to the 5' and 3' end and go for sequencing. So the DNA fragmentation can be achieved or not only from the by PCR, but through sonication, nebulization, and enzymatic way. These are sonication and nebulization are the mechanical way of DNA fragmentation, and the enzymatic way is the use of different restriction enzymes. So in this gel electrophoresis, which has been performed on agarose gel, you can see this is an intact genome, which is showing a higher molecular weight and corresponding to the DNA marker ladder, which has been run along with this agarose gel. And as you can see, the fragmented genetic material, so it almost produces a smear because the genetic material is fragmented into continuous fragments. So you can identify them. So if you have applied any particular technique, you can check it on agarose gel to find out whether you have been able to create your desired fragment for your downstream analysis. So the shared or fragmented DNA will look like this in an agarose gel. So the restriction enzymes are restriction endonuclease are enzymes which can cut within the genetic material. So endonuclease are as it is designed to cut within a particular regions, which are generally palindromic in nature and can be classified into two typical classes, which is a blunt end cutter and a sticky end cutter. So you might have heard about, learned about this restriction enzymes in your curriculum. As you can see here, a small one enzyme can create a blunt end of the genetic material, whereas enzymes such as EcoR1 can produce a sticky end or a staggered end cut. So you can use these kinds of enzymes for fragmentation of the genetic material. The next very important step is to quantify the input DNA for the library preparation and also estimate its purity. So here I will talk about a qubit fluorometer. So you can use, because use UV absorption based methods are not very useful for identifying or quantifying the genetic material for library preparation. So hence we use a qubit system where it is supplied with a I'm sorry, with a fluorescent dye that binds with the double-stranded a single-stranded DNA, and then you can accurately quantify your amount of DNA approximately present in a sample. Maybe you are estimating 100, you may require 1 microgram of DNA for your library preparation, and you will see that actual quantity of double-stranded DNA present in that sample is around 700 or 800 nanogram. So you have to adjust accordingly and it's a very useful technique and has been frequently used by different laboratories for the estimating of the input DNA and analyzing its purity. So the next important step, once you have been able to fragment the genetic material and estimate the input for library preparation, the next immediate important step is end repair. So this end repair, because the shearing of DNA or fragmentation of DNA may produce a blunt end or a fragment or overhang genetic material. So we need to repair those ends, and for the purpose of A-tailing and adapter ligation. So majority of the random fragmented methods produce double-stranded DNA with a mixture of blunt ends, recessed in the 3' end and recessed at the 5' end, with and without a 5' phosphate moiety. So these must be made uniform before the adapter ligation. So all those fragments which are still sticky or staggered end, they must be end repaired to produce a blunt end DNA fragment with a phosphorylated 5' termini. So the next uh, step is A-tailing, addition of adenine residue. So say addition of a single templated adenine nucleotide to the 3' end of the fragment before adapter ligation deters the concatemer formation of the template. So if you leave those DNA fragments, they have a tendency to rejoin and form a concatemer, and you prevent that by adding adenine nucleotide. and and because of the adapters are T-tailed, they will find soon, it increases the efficiency of adapter ligation and decreases the formation of adapter dimer formation. So several library preparation kit like kappa hyper prep or next neb next ultra 2 DNA, these steps has been combined with end repair steps. So end repair and A-tailing and adaptive ligation, this has been in a single step reaction, which ultimately saves a lot of time. So next step is adaptive ligation, where we need to uh, join some adapters at the end 5' and 3' end of your fragmented DNA for the purpose of library preparation. So the template strands receive a different adapter sequences at each end to participate successfully in the cluster amplification and sequencing reaction. So adapter ligation to the template must be as efficient as possible, but at the same time, ligation of adapters to one another must be suppressed. So once these all end repair, A-tailing, and adapter ligation has been performed, we have to remove the excess adapters from the sequencing mixture through through some cleanup process, because it can produce noise in your sequencing data. So this is a pictorial representation, whatever I have shown you in the previous slide. So once you fragment the DNA, so as the fragments are random, there must be some development of three blunt end and also staggered end cuts, and those staggered end must be repaired, and addition of 5' phosphorylation must be done. Then after the cleanup process, then A-tailing was performed. And once A-tailing has been done, we move for adapter ligations. So there are two different types of adapters, and different kits may provide you with different types of adapter. One is known as HP adapter, which binds to the T overhang at the 5' end, and it is called HP because of the enzyme the uh, which helps into the formation of this adapter, and Y adapter because of its structure. So for the HP adapter, we use helicase enzyme. And for the Y-shaped, as you can see in this image, how it looks like in a Y-shaped structure. So then you can apply a PCR enrichment, where through some limited cycle you can add these indices to the 5' and 3' end, and which helps into the downstream sequencing and also identifying different clusters. So what is indexing? Say for example, you have collected uh, genomic material from two different sites, or two different individuals, or from different individuals, or on different time point. So performing sequencing or performing metagenomic analysis one by one will produce huge cost, rather than if you can run the sequencing simultaneously by adding all the samples together in one run, it will reduce your cost. And you must be thinking, how is that possible? We have the only fear for the people of, in the molecular biology field is contamination. So basically, when you mix two different or three different samples, you have contaminated it, right? So here is the only thing. If you add, before mixing the samples all together, if you have an unique indices attached to your sample, and then after the sequencing was complete, through computation and analysis, you can through R programming or Python programming, you can write a script and send it that please segregate my data reads on the basis of indices. So if you have indices A attached to your sample, corresponding your sample A, you can easily segregate from your total pool, and then you can assemble it individually to find out the population. So this will reduce your sequencing cost hugely. So that's why indexing is important. So and then during the library preparation, libraries from different samples can be prepared by barcode sequencing. So they can be pooled and sequencing this for sequencing, and the sequences obtained from samples can be organized into separate folders afterward. So there are three different kinds of indices. So first one is single indexing. One is dual indexing, that is two different kind of uh, uh, indexes are attached to the 5' and 3' end, and unique indices, where, as you can see, different kinds of I5 and I7 indices were attached to the 5' and 3' end. So thereby you will be able to identify different samples at a time. So once everything is done, you have fragmented your DNA, you have added, you have created blunt ends, you have added 5' phosphorylation, then you have E-tailed your samples, and after then you have adapter ligations and addition of indices through some limited PCR processes. Once you have generated all these, you need to clean up. You need to identify your genetic material which has been properly processed from whatever the primers, fragmented materials, adapters. You have to remove those clusters from that. So here I have previous talk I have discussed about this solid phase reversible binding immobilization technology. So where you use an amp pure bead, where it is nothing but a silica based bead with a paramagnetic material or core inside it. And when we apply the magnetic field, once the material or the beads bind to my desired genetic material, it will be separated from all the debris. And after ethanol washing and drying and dilution, you can have your pure quantity of genetic material which can be used for sequencing. So nowadays there has been a technique which is very common. So we tag the DNA genetic material first, and first we fragment them and then we tag the genetic material. So this is two different steps. How about combining these two? And hence we have another technique called fragmentation. So there is a kit available by Illumina, which is called Nextera, and which applies the fragmentation procedure by which this is, I will go back to this slide again. So this is a pictorial representation. So they use uh, these transposons. So you might have heard of transposons, which are which can have an ability to bind the add the genetic material from one region to another region. So they have genetically engineered those enzymes and have added my desired tags, which must be incorporated into the 5' and 3' end. And when they bind to the genetic material, they introduce these tags onto the genetic material. And thereby, after PCR technique, we can add the indices to this fragmented DNA. So this is how we prepare the library, and it reduces time of library preparation hugely. So, so if you go for fragmentation for a longer period of time, or have a short period of time, both are very detrimental for your sequencing approach. As you can see here, I have shown you different concentration of the fragmentation processes. And as you can see, along with different time points and concentration, your fragmented DNA material is different. And through SPRI, you can isolate your desired amount of J fragments from the sample. So using no SPRI, as you can see, there are smaller fragments which corresponds to this peak in this M, which is represented by M here. And another large targets which are beyond my sequencing capability, which are around more than 1 kb in length. So my ideal amount of DNA fragments required for subsequent sequencing is this area, which is marked by red boundary. So to isolate that, we use SPRI cleaning technique and isolate those libraries and send it for sequencing. So what are the things we should care about this fragmentation process? So if we have too little DNA and degraded DNA from our from the input, it can lead to over fragmentation because you are adding an enzyme. So if your initial material is very less, it will chop your DNA into smaller pieces, and these small piece of DNA will not be efficient for any sequencing techniques, and your overall sequencing quality will be very low. And under fragmentation, even if you add very little amount of enzyme or the presence of any artifacts like EDTA, protein, detergent in your, and phenol. If you remember, in the last talk, I have repeatedly told you that the presence of phenol in your extracted DNA sample, if it is present, it can hinder the downstream analysis. So here it is, it may lead to under fragmentation of the DNA fragments, and it will lead to longer DNA fragments. So you must be thinking that if I had a problem with a shorter DNA fragment, what will be the problem with the longer DNA fragment? I can still go for sequencing. Yes, but depending on the type of sequencing technology you are using, longer reads might require other kinds of sequencing platform, and it might be your sequencing cost must be maybe increased. So for that particular reason, these two particular reasons, you need to care about your input DNA and carefully select the fragments which would be in between uh, 200 to 600 base pair for your subsequent DNA analysis. Next is library quantification. So this library quantification can be analyzed in manual quantification and bead-based normalization. Manual quantification is to some extent a crude method where we use a qPCR-based melting curve analysis, where you can identify your shorter fragments or your adapters if it is remaining in your prepared sample, or if you can use fluorescent dyes like pico green, which can bind to double-stranded DNA, and you can go for a bead-based normalization. So the bead-based normalization applies these SPRI-based techniques where they bind selectively to your desired fragment lengths, and they have multiple binding sites, and they can normalize your input library. So by normalization, we mean that if you can look into this image, that if you have different samples, maybe from different locations, and have different proportion of DNA as a starting material, then your sequencing reads will be also affected by it. So you need to normalize it before going it for a sequencing. So as you can see, normalization will make an amount equal. So you need to dilute the sample B depending on how much quantity you require to normalize with A and C. So that's how you do the normalization. There are techniques which are provided by the companies which help you to pooling, normalization, and sequencing. So what do you mean by library quantification and normalization? So for the Illumina, because I'm using Illumina because majority of the sequencing platforms are there now used nowadays are from Illumina. So it recommends quantifying your library using a fluorometric quantification method that uses double-stranded binding dyes. So probably you can use pico green for that. So calculate the DNA concentration in nanomole based on their DNA's amplicons determined by Agilent technology bioanalyzer trace. So bioanalyzer trace helps you to identify the exact amount of fragments which are present in your processed uh, sample, and then you can apply, say for example, the concentration of nanogram of microliter per microliter, the concentration if you have 15 nanogram of sample, and it will be corresponding to 45 nanomole. So dilute the concentrated library using resuspension buffer, as I have shown you in my previous slide. What is the meaning of normalization? And then the aliquot 5 microliter of the diluted sample from each library, and mix an aliquot for pulling this library with unique indices, and you can go for sequencing. So there are different requirements by different sequencing platforms, and with the further input volumes. As you can see, ISEC 100 requires 1 nanomole of library concentration with a volume of 5 to 25, whereas the MISEC requires 2 nanomole with 10 microliter. HiSec 2500 requires 2 nanomole with 5 microliters. As you can see, along with the advent, there are the amount of DNA required may be adjusted. So HiSec 4000 requires 2.8 nanomole of a library concentration with a volume of 5 microliters. And high-sec X requires 2.8 in nanomole with 5 microliter concentration, 5 micrometer volume. And the Nova 6 requires a little more, where it is 2.25 nanomole with 85 to 310 microliter of volume. So the library preparation, the way you choose your library preparation method can have a significant impact on your community. It can create a false positive library depending on the type of library you have selected. So if your genetic material have a higher GC content, it can inflate the bias in error profile. So these are in still development. So maybe in future you will have any techniques available where you can completely remove this bias. But even you have to adjust nowadays, you have to adjust with these biases. So if you have your, so that's why it is preferable to use a PCR-free technique for any metagenomic analysis. Other than when you use these techniques, you will find the GC reads can produce error profiles and duplication and read loss in your sample. So I have shown you a comparison of different techniques like Nextera, Mondrian, and Malbec, which have different numbers of reads and insert size and only the GC content, how it can affect their number of reads. So the PCR will introduce sequence bias. So these are the drawbacks of the library preparation and should be considered while you have planning to do that. So PCR will introduce a sequence bias in your library and should be avoided if possible. So if you have only very low quantity of DNA sample, you have to go for a PCR based amplification, because you cannot move for any a PCR free technique with a very little amount of DNA. So the most PCR free protocols, however, will require at least 200 to 2000 nanogram of the starting DNA material. So if this is not available, the user will need to require a PCR step in the library preparation. Again, the different APCR enzymes show different tolerance for the extreme GC content region and template lengths. So demonstrate different degrees of bias. So if you have a GC rich regions, those ambiguous sequences will be left as a gap in your sequencing reads. So typically amplify libraries to 6 to 14 cycles. Over amplification of your samples may create error in your rank. So that's why while introducing indices, you should restrict your amplification to 16 to 14 cycles to increase the library size and enrich your fragments. So this is what is about library preparation, and I have mostly discussed all the available techniques nowadays. And if you have any questions, you can ask.
Thank you, Mr. Roy. Spectators, if you have any question, please put it in question answer or chat box. By this time, I am having a couple of questions. So first one is, how many indices are required if I use more than 3 samples?
For using more than 3 samples, if you apply in permutation combination, if you select for one sample 2 different kind of indices, so for 3 samples you can have 6 different indices attached to your samples.
Okay. Then qubit system, you're recommending for qubit system.
Yes.
Is it really accurate? How far it is accurate over the conventional system?
Well, apart from the 260 280 based quantification, picogreen and using a qubit system is more advantageous and can have almost accurate. You cannot say it's very accurate because still you can still have an adapter dimers, concatenamers present in your sample, which can produce small a little bit of error rate. But as I have told you, that if you are expecting that your input DNA have a quality about 1 microgram, probably there must be, if the quantification will give you an estimate about 800 to 900 nanogram of DNA. So this is almost perfect, you can say.
Okay. Regarding fragmentation, how do I know my sample is over fragmented or under fragmented?
You can use a bioanalyzer. So if you have a over fragmented DNA, you will have very small fragment size. So if you can, I can show you the slide again. Just allow me to show you the slide. You can have a good
Okay,
idea
yeah.
of.
I think the next one. This.
Uh, yes. No, I had a different one. Okay, yeah, this will look
This one.
good. So as you can see here, so if you have a very small sized DNA fragment, so these area will be occupied by these. And if you have a under fragmented DNA fragment, your peak will be shifted towards this side. So you can, using, this is an image from the bioanalyzer. So you can identify your quantitative amount of or fragmented DNA present in your sample.
Another question. This is from students, I think, because the beginners, they experience some problems to detect whether the fragmentation, that is enzymatic fragmentation, is okay on agarose gel. How actually they cannot detect really, is it really fragmented or some trails are coming, how they can come.
This, I think this is not related to metagenomics, right? So this is a general question. In general, it's a question. So I would prefer, if you have your DNA fragmented, and first of all, from my personal experience, I would suggest you to build an agarose very, very thin as thin as possible to visualize your DNA. So if you concentrate your DNA, probably you will be doing 1.5 or 2 concentrated of agarose gel, then probably the smaller fragments will not be visible on agarose gel using UV. So I would recommend, if your fragment size you are expecting is around 400 to 900, you can apply a polyacrylamide gel for analyzing the fragments. So that will give you a better resolution.
So any question from any other student? Thank you, Mr. Roy. I'm expecting other questions. I think. Is there any question?
Participants, if you have any questions, please put it in the chat box or in the question answer section. It is now open for question answer session. So please, if you have any question, please put it in the chat box or in the question answer section. Ma'am, probably there is no more questions.
Okay. So Dr.
We have
Dash.
provided the feedback link and joining link for tomorrow's sessions.
Okay. Dr. Dash, do you have any thing to add?
Okay, I am here. So we will meet tomorrow. So that's fine.
So thank you, Mr. Shubhrajit Roy. We will join tomorrow again. And
Thank you.
students, you will join tomorrow, 9:30. Okay. So let us end the meeting here.
Thank you, ma'am.
Okay.
Thank you, ma'am. For all participants, please note down the feedback link for today's session and the joining link for tomorrow. It has been posted in the chat box, so it is requested to all participants to kindly take note of the feedback link and the joining link for tomorrow's session. Feedback link contains the attendance sheet